Ovita and beef and lamb genetics are not the only program doing genomics. There's also some work going on, some other programs, including the FarmIQ Primary Growth Partnership, and we've got Shannon Clarke from AgResearch speaking about some of the meat quality work being done there.
Thanks, Neville. So I'm going to talk today about utilising a high-density SNP chip for meat quality traits in the New Zealand composite terminal sire breeds in New Zealand. So just a bit about FarmIQ. So it's a joint New Zealand government and industry primary growth partnership program. Its main aim was to bring farmers and consumers together. Farmers understand how to produce animals that meet consumer preferences and hopefully receive payments based on meat quality. And for consumers are offered a premium-based red meat that consistently meets their eating quality preferences. So the FarmIQ program itself has 5 different sub-projects, and we have the product development up here. So this extensive market testing is providing better insight into consumer preferences. We have processor feedback. This is electronic tracking and extensive measurement in plants, providing data on key parameters. We have down here farm performance, best practice production systems using data capture and analysis to better align with value chain being developed. And we have a genetics program here, utilising advanced genomics techniques being used to help farmers select for better breeding of meat, yield and growth. And all of this is brought together by a FarmIQ management system. So for the genetics program within the FarmIQ, which we've been involved in, the aim is to deliver high quality genetic and genomic breeding value predictions for growth, meat yield, quality and terminal sire flocks. So lamb is a premium priced product and should be a good eating experience, so we want to develop tools to tailor products to consumer specifications. We've concentrated on the terminal sire breeds with the aim of delivering increased profitability for each of the value chain participants, being the farmer, the processor and the retailer. So there's 4 phases within the genetics program. First was the development of a high-density SNP chip. The second phase was to identify and measure eating quality and carcass yield traits in the progeny of terminal sires. Third phase was actually genotyping these individuals and generate prediction equations. And the fourth phase, where we are at the moment, is beta testing these results in a commercial terminal sire breeding flocks to ensure the system is accurate, practical and cost effective. So for the development of the HD SNP chip, this was for genomic selection, but also for broader experimental possibilities. So within the genomic selection, we were hoping that increased marker density would ensure a better marker trait association, increased prospect for successful crossbreed prediction, and it's part of a toolkit to drive genotyping efficiency. For broader experimental possibilities, we have loss of function SNPs have been included on the chip, also rare SNPs, and we're hoping to facilitate merging with gene expression studies. So this is a busy slide, but I just wanted to point out that the development of the HD SNP chip was with FarmIQ and the International Sheep Genomics Consortium. This consisted of people in Baylor, CSIRO, DPI in Australia and AgResearch. It utilised the latest version of the sheep reference genome. We had a substantial resequencing effort both at Baylor and with AgResearch. Then a whole lot of bioinformatics analysis was undertaken for the mapping of the resequenced animals, SNP identification and SNP filtering to choose the SNPs for the chip. So the HD SNP chip contains oligoprobes for 685,000 SNPs. With these we have nearly 80% of these are of equal spaced across the genome. We have the previous 50k SNP chip SNPs on here as well. We also have about 2% SNPs accessible by a newer genotyping technology, genotyping by sequencing. This allows us to have both forward and backward capability. We also have about 8% of functional or putative functional SNPs on here, or loss of function SNPs. So following the manufacture of the chip, we had 87% actually passed manufacturing QC. In the New Zealand population, we have just over 600,000 SNPs with a mean call rate of 99.7%. and we have 589,000 SNPs that are greater than the threshold of 98% of the animals have to have the genotype. For the trait investigation, first wanted to identify and measure eating quality carcass yield traits in the progeny of terminal sires. Our animals were processed through silver and farm plants. We utilise a system that's automatically tracking carcasses based on the EIDs. Our eating quality indicators were meat tenderness, pH, marbling, colour and colour stability. And we've been working with 11 New Zealand flocks that produce Texel terminal sires or composite type genetics from Suffolk, Texel, Poll Dorset breeds, and most of these are part of the Focus Genetics network. So just an example of the numbers that we have in the program. So we have 3 progeny tests and 2 monitor farms. We have 839 unique sires. We have yield measurements from progeny of just over 15,000. Of these, we have 10,000 that have meat quality measurements, and we also have been investigating NIR as a proxy for some of these measurements. So just a summary of the genetic parameters, because Louise gave a great talk on Monday summarising all of this. So the traits are heritable, the meat quality and carcass traits especially. We've got variation in growth with a coefficient of variation of 14% of the mean, and this is even greater for meat yield and quality traits. So phase 3 is the actual genotyping analysis and mBV predictions. So our genotype resource we utilised for this was 10,000 animals born from 2010 to 12, all being genotyped using this HD chip, including the size. From the born 2012 and 13 genotypes, we've got a combination of low density and high density chips. A phenotype resource, 85 traits available. We've focused on just over 1,000 phenotype records, mainly looking at meat quality. The model that was used, and this is work that's been done by Michael Lee, we've got fixed effects, flock, year of birth, breed, mob and sex. For meat quality, we've also got a covariate of carcass weight. The genomic relationship matrix utilised 20,000 autosomal SNPs and was used to fit a polygenic model. And the residuals used a test for association with each marker with simple linear regression. And just an example of this, so each colour represents a chromosome, with each spot representing a SNP marker, and the significance up on the y-axis. So for tenderness, marbling, colour, fat depth, eye muscle and carcass leg length, you can see there's quite significant regions of the genome for these traits. Just to show that for weaning weight and pH, we don't see really any of these really highly significant areas as you would expect for these traits. So just for the accuracy of genomic selection, so this was just a preliminary look at this. It utilised 100,000 SNPs were randomly chosen from the chip to do this work. In the light blue above here, we've got just utilising one genetic group in training, and in the dark blue down here, we've basically utilised all the genetic groups. So in the training, these are the numbers in the training set for this, and here they've been reduced. But over here you can see the accuracy is quite good within breed, but it also holds up across the breeds as well. So phase 4 is actually testing in commercial flocks. We want to test the results in terminal sire breeding flocks to ensure the system is accurate, practical and, probably most important, cost effective. This is assisted by using a low density SNP chip, which has currently got just over 5,000 SNPs on it. We're currently building a new one that's going to have just over 15,000 SNPs. Breeders have been using this chip to assess their young ram lamb crop in early 2015. So just over 2,000 born utilising part of an existing DNA parentage, or an enhanced DNA parentage as we like to call it, and using industry mating it has just occurred. Just want to point out that this work complements other approaches to improving eating quality, including enhanced pre and post slaughter management. So in summary, meat quality and eating traits are heritable. Genomic prediction accuracy from DNA is up to 50%. Looks like we've got some loci with major effects. We can predict across breed. probably only explain moderate proportion of variation, and we may have to fix or discard if effects are both positive and negative. And just acknowledgements from the team at Ag Research. This work's driven by John McEwen, Wendy Bain in particular, who organises and collection of all that phenotypic data, and Michael Lee, who's been doing the GWAS and genomic selection analysis. Thank you.
Thank you, Shannon. Are there any quick questions? Peter?
Yeah, Shannon, when you mentioned that you can predict across breed, does that mean that you can train in one breed and predict in another, or does that mean that you can train in a mixed breed population and then predict in populations with those breeds?
Could you repeat the question? Sorry, Shannon.
So Peter has asked if, when I said we can predict across breeds, whether we are training with all the breeds in there and then also predicting across into another breed, or just keeping all the training population together and predicting. So with that, it was my understanding that all of them were used in the training set, and then when used to actually predict across the breed for the other ones. But Michael might want to comment on that.
Okay, so it's basically a within breed
Yeah.
training set. So you wouldn't probably use that same calibration to predict across to another breed.
So you can't train in one breed and then predict in
the
another
other.
breed?
Yeah, I would probably not.
Okay.
Depends
I think that statement was in context of the GWAS results.
Yeah.
So I was talking about if you've got a single SNP, then it may well be variable in more than one breed. So,
Okay.
Okay, we'll save any more questions for the panel discussion. Thanks again, Shannon.